Can somebody confirm you can hear me?
Yes.
Thank you. All right, before we get going, are there any questions? All right, let's begin. Oh, let me show my screen here. There, you should be seeing my screen now. Today is January 27th, and we should be finishing chapter 3 and starting chapter 4 today. I think we'll succeed in doing that. Let me warn you, there is another quiz this week. On Wednesday, there will be quiz 2. Quiz 2 will cover lab 3 and 4, and chapter 3 and the end of chapter 2. I sent somebody the page number where I was breaking off on, I guess that was chapter
You
2.
you had said that it was going to be from page 36 on.
Thank you. Yeah, I know I've got an announcement about that, so thank you. So chapter 2 will begin on page 30, what was it, 36?
Correct.
Okay. All right, there is a lab today, lab 5. In the lab, I'll talk about the grading that I've got done. Let me say that I have one thing up. Let me see if I can find it. One thing up that I finished last week. I think it was after Thursday class, but I might have forgotten just to mention it on Thursday. I think it's under week 3. Maybe it's week 4. Oh, there it is. Week 4 lecture. I've got an environmental scan. You can click on this link. It will not open in Canvas, but it will give you a link. Oh, I take that back. It is opening in Canvas. Anyways, you wanted to see an environmental scan. A student asked that. I think 2 students actually asked that uh, when I tried showing you before. And I was able to show you the monitor and how you move it, but you couldn't see unless the monitor was in front of me because of the background image. And I did mention that it was just too difficult to remove the background image from the Zoom. Um, it took me about 5 minutes alone just to log in and verify my existence because I had to verify my existence twice, and that is Zoom will send me back to the college page. And to get into the college page, I have to verify my existence, and then I have to verify my existence with Zoom. So, like I said, it took me 5 minutes to log in alone, and then it took me another 5 minutes to figure out how to remove the image and get it done. But you can see there's no background image here. This is a table where I'm showing you how to scan your table. Any question about any of that? All right. This isn't needed too much anymore. I realize that because after the last quiz, 90 to 95% of you were actually showing me your environment correctly, meaning the area around your computer or where the keyboard sits. Any questions about anything before we get started? All right. I have that we had talked about the basis of Gram staining, why some stains stain purple and one stains pink. What's causing this differential staining? It is the thickness of the cell wall. We haven't covered this, have we? Anyone? All right. I'll just assume this is where we are. Um, that is, the gram-positive bacteria have a thick peptidoglycan layer, and the crystal violet iodine complex gets stuck in that thick peptidoglycan layer. And that's why the cells remain purple and the gram-positive cells then stain purple. The gram-negative cells have a thinner peptidoglycan layer in their cell walls. They also contain a liposaccharide layer outside as part of the cell wall. And because that layer is thinner in the peptidoglycan, the crystal violet iodine complex washes out of it. So here, let me blow this slide up so you can see it better. There, the purple, we see the cell wall. The yellow underneath is the cell membrane. And it's a thick peptidoglycan layer in the gram-positive cells, much thinner in the gram-negative cells. And then we're looking at the artist rendition here of the cell wall. It's only showing you 3 layers of peptidoglycan. Let me remind you that in a gram-positive cell, we have something like 30 layers to hundreds of layers of peptidoglycan, not just 3. And there's the plasma membrane. In the gram-negative cell, we have a lipid layer outside the peptidoglycan layer, and this is all part of the cell wall. This lipid layer of the cell wall is actually called the outer membrane of the cell wall. The thinner peptidoglycan layer, which this has only 1 layer, remember that it's actually between 3 layers of peptidoglycan and 20 layers of peptidoglycan, but the artist just shown 1 layer. Because this layer is much thinner than the gram-positive cells, then the crystal violet iodine complex washes out of the gram-negative cells. And this outer membrane actually gets destroyed in the decolorization step of the gram cells because the alcohol will dissolve the lipid layer. So the crystal violet iodine complex forms in all cells, both gram-positive and gram-negative, after the addition of iodine. But this complex is larger than the crystal violet or the iodine alone, and it gets stuck in the gram-positive cells because it's simply a thick peptidoglycan layer. The cells therefore remain purple. However, in the gram-negative cells, the crystal violet iodine is washed out in the decolorizing step because the peptidoglycan layer is just thinner. And like I said, the outer membrane of the cell wall, the lipopolysaccharide layer, it's disrupted, it's dissolved by the alcohol wash of the decolorizing step. So here we see gram positive cells and gram negative cells, the pink cells. Any question about any of that? All right, the gram stain, you'll frequently see something like this. How do you score this? These look gram negative there, and then these look gram positive there. These are bad regions of the slide. These are good regions of the slide. You want to use the good regions of the slide to score your gram stain. If you look there carefully, you can see that these are not actually cells. These are crystals of crystal violet, meaning when you make crystal violet, sometimes the solution starts to crystallize, and that's a crystal of crystal violet. And when somebody made this slide, they picked up the crystals and put it on the slide. And you can see that the cells right next to the crystals, these right there, and these right there, are much darker than the cells away from the crystal of crystal violet. And so you don't want to score the cells in the bad region of the slide. Any question about any of that? The gram stain is actually kind of tricky. And if you were to actually do it, some of you would be pulling out your hair like this student is doing here. Most consistent results of the gram stain are performed on young growing bacteria cells. So if your cells are older than 2 days old, you should streak them out and use young growing cells that are 2 days old or younger. You do not get good results when you stain old cells. Some bacterial stained cells stain poorly or not at all. It's mostly the mycobacterium cells that stain poorly or not at all. The reason is the mycolic acid in the mycobacterium. It's a genus and family of bacteria. It has mycolic acid in it, and the mycolic acid in the cell wall does not allow the water soluble molecules to come through the mycolic acid. And the gram stain is dissolved in mostly water, and so it does not stain the cells very well. However, if you do use cells of less than 2 days old or younger, the cells do stain gram positively, which is what they are, gram positive cells. But if you have older than 2 days old, some of them may not stain, or they will even stain gram variable. I'll talk about that in a minute. So the results of the gram stain are not always as clear cut as theory suggests. If you were due to the unknown project, we'll do that project later this term, and you are actually due to the gram stain. Like I said, some of you would be pulling out your hair because it is tricky. Make sure you always stain 2 days old cells or less, and then usually you won't have problems. The structure of the gram positive and gram negative cells not only affects the staining in the gram stain, the structure of the cell wall also um, pertains to how the cells respond to some antibiotics. Gram-positive bacteria tend to be killed easily by penicillin G, cephalosporin, and many detergents. And that's because penicillin G, cephalosporin, and many detergents go right through the gram-positive cell wall. They're water-soluble molecules. They go right through the gram-positive cell, and then they can kill the cells. The gram-negative bacteria are more resistant to penicillin G, cephalosporin, and many detergents. And that's because they cannot get through the lipopolysaccharide layer of the cell wall, meaning the outer membrane of the cell wall protects these cells, the gram-negative cells, from penicillin G, cephalosporin, and many detergents. Any question about any of that? Now realize we do have antibiotics to kill gram-negative cells. We even have members of the penicillin family, like ampicillin, the third or fourth member of the penicillin family to be discovered. They actually chemically modified it, but one of the very early penicillin members of the family actually works on gram negative as well as on gram positive. Penicillin G, the first member of the family to be discovered, does not work on gram negative cells, and that's because the penicillin G cannot get through the outer membrane of the cell wall. Any question about any of that? All right. You will not be examined on special stains like the capsule stain, but I do want to talk about the capsule stain because some of you will need to do the capsule stain in your unknown project. The capsule stain is used as a stain for staining the capsules around cells. You cannot see the cell capsule in a normal staining procedure. In fact, in the gram stain, the cell capsule is washed off the cells. You need a very gentle staining procedure, and then also a special staining procedure to make the capsule show up. Here we're using a two stains, actually, one that stains the background of the cell, another stain that stains the cells themselves. And as you can see, the capsule does not stain, making the capsule appear very distinct. This is usually done with a negative stain, meaning you simply stain the cells. Usually you stain the background first, and then you stain the cells, or you stain the cells first, and then you add the stain, and you just leave it on the slide, and you look at the cells, and then you can see the capsules. Here is India ink, one of my favorite negative stains, because it's so simple. You just use one stain, and this stain does stain the cells lightly. They're sort of a light gray color, and then it stains the background of the cell very dark, almost black. And the cell capsule does not stain. And this is what a negative stain looks like, by the way, where the cells are either dark or light, and it sort of looks like a Polaroid negative. Any question about any of that? All right. I think that's it for this lesson. Any questions about chapter 3? If not, let's go on to chapter 4. Chapter 4, the functional anatomy of prokaryote and eukaryote cells. Let me warn you, I do not usually talk about the functional anatomy of eukaryotic cells because you've already had that in your preliminary course, meaning Biology 160, if you took it at Clark College. You should have had the anatomy of eukaryotic cells. I will provide the slides, but I do not usually spend the time covering the eukaryotic cells. We will talk extensively about the anatomy of prokaryotic cells. This slide is showing you the major goals and a rough outline of chapter 4. Know the terms. What's a coccus, a bacillus, a spirilla cell? What is strepto, like streptococcus, staphylococcus, diplococcus? What's a tetrad? What's a sarciniae? What is a vibrio? What is a spirochete? What is the glycocalyx? What is a flagella? What are the axial filaments? What are fimbriae? What are pili? What is peptidoglycan? What is an endotoxin? And what is the selective permeability? What is diffusion? What is facilitated diffusion? What is osmosis? Active transport? Tonicity? What is a plasmid? What are inclusions, also called inclusion bodies? What are endospores? And what is the endosymbiotic theory? The one thing I will talk about eukaryotic anatomy is the endosymbiotic theory. All right, 2. How are gram-positive and gram-negative cell walls different? We just talked about that. We'll go into it in a little more detail in this lesson. And then lastly, describe the importance of taxes in bacteria. Any question about the overview and the rough outline? Remember, to learn all of the goals, you need to go and take a look at the objectives to the lesson, which you can find in the study guide. It's just this very start of the study guide will be the objectives. Any question about any of that?
Would it be safe to assume that the objective is going to be where majority, if not all, of the quiz questions will relate to or
Yes,
come
if
from?
you know the objectives, you should be able to answer almost all of the questions, at
Okay.
least the objectives in the lecture. Obviously, there will be questions also from the lab.
Okay.
I removed the objectives here because I wasn't consistent. And so this lesson had an objective separate from the study guide. I've now put the objective in the study guide. And then all of the study guides just have the objective at the start of the class. And so I really should remove that. I just kept it because this is the way it used to be. All right, let's go back to the lesson. So this is showing you a comparison of prokaryotes and eukaryotes. The Greek word prokaryote literally means pre-nucleus. Strictly speaking, it means pre-kernel, meaning karyotes is kernel, but they're using the word kernel for nucleus. And then eukaryotes literally is Greek word for true nucleus. True, karyotes, kernel for nucleus. Prokaryotes, especially the bacteria, have one circular chromosome. We'll make a circle like that. And the beginning point is the same as the ending point, meaning the chromosome is in a circle. The chromosome is not in a nucleus. Eukaryotes generally have, actually, as far as I know, all of them have linear chromosomes, like the X chromosome down here, whereas a beginning point and an ending point, and the beginning point and the ending point are separate points. They're not the same point. The chromosome is not in a nucleus in a prokaryote. It is, the chromosomes are in a nucleus in a eukaryote. The DNA of bacteria is not associated with histone proteins. Histone proteins are a protein that the DNA wraps around in a eukaryote cell. So it's the main protein in the chromosome, but there are other proteins in the chromosome. And it is true that bacteria chromosomes do have proteins. They just don't have the histone protein. Prokaryotes do not have any organelles. I'm talking about membrane-bound organelles like the chloroplast, the mitochondria, the Golgi body. And eukaryotes do have membrane-bound organelles. Bacteria do have peptidoglycan in their cell walls, and actually only bacteria have peptidoglycan in their cell walls. The archaea, or the archaea bacteria, do not generally have peptidoglycan in their cell walls. Eukaryotes have no peptidoglycan in their cell walls, and if you recall, animal cells do not even have cell walls. Prokaryote cells divide mostly by binary fission, where one cell will simply split down the middle and divide into two cells. Eukaryote cells divide by mitosis. And hopefully you learned that in your preliminary biology class. Any question about any of that? All right. So we're first going to talk about the prokaryote cell shapes and their cell arrangement, also called the cell growth arrangement. We'll talk about cell structures. We'll talk about the movement of materials across membranes, like the plasma membrane. We will then switch gears and stop talking about prokaryotes and talk about eukaryotes. And all I'm going to do is really show you the difference between a plant cell and an animal cell, and that's it. And then we'll switch and talk about the endosymbiotic theory, the theory of how eukaryote cells kind of came into being and how they got the mitochondria as well as the chloroplast. All right, the prokaryote cell size and shapes. The average size of a prokaryote cell is something from 1 micrometer to 10 micrometers. It's really closer to 0.4 micrometers to about 10 micrometers. And the average size is really 1 to 2 micrometers for the average. Oh, we're talking about the length. That's why it's 1 to 10. All right, sorry. That's the diameter I was giving you. And the length, the longest prokaryote cell is about 10 micrometers long. The bacteria have basically three basic shapes. A coccus cell shown here, which is kind of a round ball-like shape. The bacillus shape here, which is also called a rod. And then the spirals, or the spirilla, shown here. And that looks sort of like an S. All bacteria, or almost all bacteria, are one of these three shapes or some combination of these three shapes. Like when we talk about Coccobacillus, that is a rod that you just shrink and shrink and shrink, and it has rounded edges. And when you get it small enough, it looks kind of like a coccus, an elongated coccus. No longer looks much like a rod. And that we call Coccobacillus. A Coccobacillus, the point is, it's in between a coccus and a bacillus, so it is based on these three shapes. And most cells are based on these three shapes. Any question about any of that? We do have different types of spirals. There's a vibrio, which you can call a half bent rod, or you can call it a half of a spiral. So there's this spiral, take a half of it, split it down the middle, and then you have the vibrio. There are some bacteria, the species actually are vibrio. Um, Vibrio chloride? Now, what the heck is that called? Uh, Anyways, there are some species, not very many, that are actually vibrio. There are a number of species which are spirals, spirillum, or spirochetes. The difference between a spirillum and a spirochete is a little bit more than just the number of spirals. The spirillum is simply one simple S. The spirochete has multiple S's in it, meaning it's bent many times. Another difference is the spirillum is also more rigid and inflexible, and the spirochete is less rigid and much more flexible than the spiral. Any question about types of spirals? And remember, you could call the vibrio a half a bent rod as well as a half a spiral. There are some prokaryotes that have an unusual shape. There's the Stella bacteria, they're actually star-like shape. There's an actual electron microscopic image of the Stella. And then there's the Archaea that is actually a rectangle. They call it a square, but it's more of a rectangle. The Haloarcula, it's an archaebacteria. It's actually a rectangle, and there's an actual electron microscopic image of it. Very rare, these number of cells. And then there are a few bacteria species which are pleomorphic, meaning the cells take on many different shapes and sizes. Most bacteria species, in fact, the vast majority of bacteria species, are monomorphic, where the cells have a single shape. Like these, all of them are the same shape. The same with the stella, all of them are the same shape. The same with the staphylococcus, the spirochetes. So most of the bacteria species are monomorphic, having a single shape. A few are pleomorphic. Most of the pleomorphic species are pleomorphic because the bacteria does not have a cell wall. The cell wall tends to give the bacteria its shape. And so the cells that lack the cell wall take on multiple shapes. Any question about any of that? We'll talk about a family that lacks a cell wall. And this family tends to be pleomorphic. To my knowledge, all the species are pleomorphic. I don't know all the species though, but to my knowledge, all of them are. The mycoplasma are the pleomorphic family of bacteria, and they lack a cell wall. All right, so we've talked about the general bacteria shapes. Let's talk about the prokaryote cell arrangement, also called the growth arrangement or the cellular growth arrangement. When we're looking at cocci, you can have a singlet cocci like this one here, or let me blow this up. So there's a singlet cocci there. Um, Actually, I'm not sure that's a singlet right there. That could be a diplococci, but that might be a singlet. So you can have singlets, and you can also have diplococci. Clearly, that's two diplococci. Those are two. Those are two. That looks like a group of them. Well, there's diplococci right there, too. And there's the artist's rendition of a diplococci, where simply two still cells are stuck together. Two cocci, in this case, are stuck together. You can also have streptococci, and the cells stick together one after another, forming a string. And there we see a streptococci ring right there. And there's another one there. It's over by that one, but the plane of division, you don't really need to know this. On a diplococci or a streptococci is always the same, meaning the cell will divide this way, and then you get two going that way, and then this one will divide this way, and then we have this one, and then the next one, and it just continues the streptococci. You can also have a tetrad, and here the cell divides on two planes. So first it will divide this way. Come on. So you got one cell here and then another cell here. And what would that be? Let's say this one here and then this one there. It's kind of hard because this is a little different angle than that is. And then it divides on this plane. And that means this cell will divide there and that cell will divide there, making the tetrad. Four cells stuck together. You can actually see the artist's rendition here. The trouble is there's so many of them stuck together that it's hard to see. But there's tetrad 1, 2, 3, 4. You have to ignore all the other cells around it, but there's a tetrad there. The sarcenia divides on three planes. So this one here, that one there, and then that one there. And that gives the cells 8 cells stuck together. 1, 2, 3, 4, 5, 6, 7. And then the eighth one you can't see because it's hidden by the other cells. It's back there someplace. And you can sort of see a tetrad right there. Obviously, you can only see some of them. And then there's so many cells here, you can't easily see the sarcenia. Anyways, note the planar division. And then with the cocci, you can also have a staphylococci. Here the cells just grow as a clump, and that sort of looks like a clump of grapes. If you were to put a stem right here, that could be a clump of grapes. And actually, that's what staphylococci means. Cocci growing in a clump looking like grapes. That's what the term actually means. And there you can see actual staphylococci, where the cells are just clumping together, growing in a clump. Any question about any of that? For the bacilli, we also have cell growth arrangements or cell arrangements. You can have singlets, so single bacilli, and there's a single. There's another single. You can have diplobacilli. There's a diplobacilli right there and right there. Note how the diplobacilli are where the cells shape long ways this way and that way, so that the small portion is stuck together and the long portion is stuck outside. That's what we mean by diplobacilli. Same with streptobacilli. it's the same shape. You're just getting multiple cells stuck together. And here we actually see streptobacilli stuck together. One, two, three cells. There's probably a fourth. That one's hidden though. One, two, three, four. This sort of looks like a sausage link. If any of you remember the old sausage links, where the sausage were stuck together in a link like that. I don't know why the author put Coccobacillus here, because this is not a cellular growth arrangement. So don't get confused on that. This slide came from Benjamin Cummings, Pearson, Benjamin Cummings, and Coccobacillus is a cell shape, not a cell growth arrangement. So it should not even be on this slide. Okay? And there you can see he's talking about Coccobacillus, which you just have a rounded rod, meaning it's not square on the edge, it's not pointed on the edge, it's rounded. And then you shrink the size of the rod, and that looks like a Coccobacillus. And there's a Coccobacillus right there. That one almost looks like a cockeye. It's so small. And then these right there look like really small rods. There is another way you can have the streptobacilli stuck together. It's called a palisade, shown right there. And here the cells are stuck on their long edge, not on their short edge. And then they look like this. Let me end the show here. If I can get paint to go, there we go. Must be Mozart's birthday today. It's awfully small. Pull it up. Come on. Unable to grab that one. There we go. I'm not going to be very good at drawing this, but let me try. The Palisade is named after the Palisade fence, which, if you read Mark Twain, Tom Sawyer was actually painting the Palisade fence. He's trying to convince Huckleberry Finn that it was a fun job. And so Huckleberry Finn started painting the fence for Tom Sawyer. Anyways, that's one place you could read about Palisade fence. I'm going to stop drawing it here. And then this is the ground here. And that used to be a very common fence, especially in apparently the 1800s, but even the early 1900s. You can still see it today. In fact, there's a palisade fence outside my window here, but I can't really show it now because I think it's dark. Yeah. Anyways, the bacteria actually get stuck together like that, and that's called the palisade, named after the palisade fence. Any question about any of that? You should also note that bacillus has two meanings. If you capitalize the B in bacillus, then we're talking about the family Bacillus. It's a family of bacteria that actually make endospores. We talked about that in the last lab. However, if there is no capital on Bacillus, then it is the shape of the bacteria. And there's a single Bacillus, the shape. So small case B, it's the shape of the bacteria. If it's a capital B, we're talking about the family Bacillus. You can also not get confused with the term Streptococcus, which is a family, Streptococcus, of bacteria. So it's a capital S, Streptococcus, and then Streptococci, which actually is spelled differently. This is usually lowercase, and then it's Streptococci, which is the bacteria lining up in a string. So don't get confused on those two. Any question about any of that? All right, let's talk about some typical structures. Bacteria. Every structure shown in red is one that you'll find in all prokaryote cells. In fact, most of them you'll find in all cells, like you can find cytoplasm, the ribosomes, the plasma membrane, in all cells. You don't have a nucleoid containing region, where the DNA is, in a eukaryote cell, but you do in a prokaryote cell. So what you could say is you have a DNA in both the prokaryote cell and the eukaryote cell. Of course, the DNA in a eukaryote is not in the nucleoid region, it is in the nucleus. The nucleus has a distinct boundary between the cytoplasm and the nucleoplasm, and that's the nuclear membrane. Prokaryote cells have no distinct boundary between the cytoplasm and the nucleoid containing region of the cell. The nucleoid containing region of a prokaryote cell is simply any region of the cytoplasm that has DNA in it. And wherever the DNA floats to, that would be the nucleoid region. And the DNA is just floating in the cytoplasm. So wherever the DNA is, that's the nucleoid region. Wherever the DNA is absent, that is not the nucleoid region, that is the cytoplasm. And I guess strictly speaking, the nucleoid region is part of the cytoplasm. It just stains differently, which you can sort of see here. The nucleoid region stains differently than the cytoplasm. It even looks different in the electron microscopic image. That's the nucleoid region. It's because the DNA just picks up stains a little differently than the cytoplasm. Some other region of the cell, we'll start there. Some bacteria have a pilus. We'll talk about that in a little bit. The pilus is used for sexual reproduction. Some bacteria have a capsule. Remember, the capsule is external to the cell wall. The capsule is external to the cell. Cytoplasm and ribosome is in every cell. The ribosome is a large structure in the cell made of RNA and protein. And it's so large that the ribosome actually never goes into solution, meaning it stays out of solution. And there you can see the dots in the cytoplasm of the ribosomes. The ribosomes are the site of protein manufacture. And if you can look over here, you can see the dots in the cell. That's of the ribosomes. They're actually smaller than shown here, but you have to show it. So the cell wall is the most external part of the cell if the cell has a cell wall, because the capsule is considered external to the cell. The cell wall is the last part of the cell. Most bacteria do have a cell wall, but not all bacteria have a cell wall. And of course, animal cells do not have a cell wall. Plasma membrane, all cells have a plasma membrane. It is what controls what comes into and out of the cell. So in a sense, it is the boundary of what is the cell is. Just the cell wall is considered a part of the cell. So you have to remember the cell wall is the last part of the cell. And the cell plasma membrane is internal to the cell wall. We already talked about the nucleoid containing DNA, a plasmid. Not all of the DNA can be in the bacterial chromosome. Sometimes DNA can be in a little molecule that we call a plasmid. We'll talk more about that later. Some bacteria have flagella, or if there's only one, a flagellum. If the bacteria can swim or move, that means it has at least one flagellum, if not multiple flagella. We're talking about a species of bacteria here. And that's because the flagella is the only structure that prokaryote cells have to move. It's not true of eukaryotes. Eukaryote cells may move by the flagella, but they could move by two other means, and we'll talk about that later. Fimbriae or hair-like structures on the bacteria cell. Not all cells have fimbriae, but some do. We talked about the plasma membrane, the cell wall, and the capsule. All right, the last thing I want to talk about is an inclusion, also called an inclusion body. An inclusion is material that the cell wants and stores. Usually the material comes from outside the cell wants it, it brings it into the cell, and then the cell stores so much of it that the inclusion comes together and then precipitates out of solution, meaning it actually precipitates out of the cytoplasm, forming a body we call an inclusion in the cell. And there's an inclusion there. Any question about any of that? An inclusion can be made up of more than one thing. It depends on what the cell is storing to be the inclusion. Let me move forward here. Well, we'll talk about it later. All right, let's talk a little bit more about the glycocalyx. The glycocalyx is the capsule that I talked about earlier. And there are three types of glycocalyxes: the capsule, the slime layer, and then the extracellular polysaccharide. It depends on how the glycocalyx is arranged and what the contents of the glycocalyx are to determine whether you have the capsule, the slime layer, or the extracellular polysaccharide. The glycocalyx is secreted by many prokaryotes, but certainly not all. I would say the majority of prokaryotes do not have a glycocalyx. It is outside the cell wall, so it's considered external to the cell. It's usually sticky, and there are three types. The capsule, which is a neatly organized glycocalyx. It has other molecules in it besides water. Water is, of course, the main molecule in the glycocalyx or the capsule, but realize that water is the main molecule in the cell, so it doesn't really mean much to say it's the main molecule in the glycocalyx. But the point is that a capsule has other molecules in it. It's not mainly just water. The slime layer, on the other hand, is unorganized, less organized than the capsule. It's very loose, loosely attached to the cell. And then the slime layer, the main molecule, is water. So it's largely water, not 70% water, but 95% or greater. All it is water. You cannot see the slime layer in the light microscope. To see the slime layer, you have to use an electron microscope. You can see the capsule in the light microscope. The capsule is important for some pathogens because the capsule can prevent phagocytosis of the bacteria cell by white blood cells. For example, Streptococcus pyogenes, the bacteria that causes strep throat, it's very difficult for the patient to throw off because the bacteria has a capsule, and that prevents phagocytosis of the bacteria by white blood cells. All right, the third type of a glycocalyx is the extracellular polysaccharide. You find these in biofilms. A biofilm is a film secreted by cells And initially, the secretion is a capsule, and then the capsule starts coming together by many different cells, creating the biofilm. We'll only talk about biofilms a little bit in this class, but one example of biofilm is the plaque on your teeth. It's made by bacteria, and they start out making a capsule. The capsules start growing together, and then we have the plaque on the teeth. Any question about any of that? The importance of a biofilm is if the bacteria is inside a biofilm, it's harder to kill the bacteria. Even with antibiotics, it's harder to kill all of the bacteria than if the bacteria do not form a biofilm. All right, any questions about the glycocalyx? The flagella is used for locomotion in bacteria as well as archaebacteria. If the bacteria can move or swim, then it has at least one flagellum, if not multiple flagella. It is largely outside the cell wall, but there is a portion of it inside the cell wall. There's even a tiny portion of it inside the plasma membrane, but the majority of it is outside the cell wall. And it's sort of like whip-like structure made of chains of flagellum, that's a protein. And the cell actually whips sort of like a whip. And it whips this way, and that'll propel the cell that way. Any question about flagella? There are four basic arrangements of flagella. You don't need to know this for the quiz. I won't ask you any of this for the quiz, but you do need to know it for the, oops, that's the wrong one. Let's see where I was. You do need to know it for the lab. There's a monotrichous flagella, and this bacteria has one flagellum on one end, called monotrichous. You can have amphitrichous, where you have two flagella, one on each end. And then there's lophotrichous, where you have multiple flagellum on each end. And then there's peritrichous, where you have multiple flagella all over the bacteria. Any question about any of that? All right. If the cells are motile and they are a prokaryote, it's because they have flagella or a flagellum to either run or tumble or swim. The movement of bacteria towards or away from a stimuli is called taxis. And in bacteria, we have two types of taxis, chemotaxis and phototaxis. Chemotaxis is where the bacteria move in relationship to a chemical. So let's say here, if there's a chemical here and the cells want to move away from it, they would then start swimming this way. There's also phototaxis, there's a light source here, and these cannot be a photosynthetic plant because they're actually swimming away from the light source. So they're responding to phototaxis, to light, and they'll either swim towards it. Usually most photosynthetic organisms will swim towards the light, but a non-photosynthetic organism could swim away from the light, and this one's swimming away from the light. Chemotaxis, that means they respond to a chemical, and they just follow the chemical gradient. If it's swimming away from the chemical, like as a toxin, then they swim away from where there's higher concentrations of the of the toxin. And so they'll swim in the opposite direction down the concentration gradient where there's fewer and fewer chemicals of the toxin. Any question about any of that? All right, let's talk about the axial filaments, also called the endoflagella. I usually use the term endoflagella rather than axial filaments, but they do mean the same thing. You find endoflagella in spirochetes and in spirochetes only. The endoflagella is anchored at one side of the cell and it is inside the cell wall. Let me blow this up so I can show it to you. Blowing it up real big. So if we were to take a cross section here, the light pink is the cell wall. Well, it's actually the light pink is the outer sheath of the cell wall, and then the cell wall is the purple and the pink. Remember, the outer membrane of the cell wall is part of the cell wall. The axial filaments are in between the outer membrane of the cell wall and then the peptidoglycan. So the axial filaments or the endoflagellum are inside the cell wall and they just wrap around the cell like this, wrapping like that. And there you can see it a little better, wrapping around the cell like that. Any question about the endoflagella? The endoflagella cause the cell to rotate in a corkscrew-like movement, making the cell move like a corkscrew going through cork. Let me see if I've got that now. There it comes. Well, I don't know why it's so slow. Boy, this is really slow. There you can see it moving in a corkscrew-like motion. And you can see, oops, let's go backwards, that to move in the opposite direction, it just starts rotating the other way. So it's moving this way now. It's about to switch. It's going to move this way. There, it's moving that way. And then look here. See there? That's how flexible the cell was. It actually bent on itself right there, starting to bend on itself, and then stopped moving north and south. And now it's moving east and west. Any question about the spirochetes and their corkscrew type movement? There it is moving this way. It's thought that this corkscrew type movement can actually allow certain spirochetes, like syphilis, to actually burrow through the mucous membranes. And then it can burrow through tissue once it gets inside the tissue, too. Shorter than the axial filament or the endoflagellum, and also shorter than the pili, are the fimbriae, which are hair-like structures attached to the cell. The fimbriae is attached to the cell wall, but most of the fimbriae is outside the cell wall. The fimbriae is shown in yellow here, and then the cell wall would be the dark purple, blue purple, and then I think the cytoplasm is the reddish purple. So the fimbriae are shorter and straighter than flagella, and the fimbriae are used for attachment to a surface. They are not used for swimming. or movement in by the bacteria. Some human pathogens have fimbriae, and then the fimbriae attach to our cells. Any question about fimbriae? All right, let's talk about the pili, also known as the sex pili. It is a long, hollow tube, longer than fimbriae, that comes out of this cell here. Let me blow that up. So you can actually see fimbriae on this cell here, and it's made by this cell, and it's a long, hollow tube. The fimbriae, excuse me, the sex pili actually goes out to this cell here, and then literally skewers that cell. The tube is hollow, and that allows a plasma then to come from this cell, move through the sex pili, and then move into that cell. So the sex pili is used to transfer DNA from this cell into that cell. Any question about the pili or the sex pili? All right, let's talk about the prokaryote cell wall. It is complex, semi-rigid structure responsible for giving the cell shape. For example, if the bacteria is a cocci or a bacillus, it has that shape because of the cell wall, giving it that shape. The cell wall is semi-rigid and it protects the cell membrane, protecting the cell from osmotic lysis. What happens is water can move into the cell. That will cause the plasma membrane to expand. The plasma membrane can only expand up until it touches the cell wall. Once the plasma membrane touches the cell wall, it cannot expand any further, and that limits water from moving into the cell. That prevents the cell from lysing when the cell is in an aqueous environment, when there's more water outside the cell than inside the cell. Like a red blood cell, which has no cell wall, you put it into a beaker of water, and the water will move into the cell, causing the cell membrane to expand until the cell will puff up, sort of like a donut. And then it continues water moving into the cell. And what happens is the cell membrane can't expand anymore, and it just pops, bursting, and that will cause the cytoplasm to leak out, killing the cell. The cell wall is located outside the cell membrane. The cell wall can contribute to the pathogenicity of some species. The main molecule in the cell wall of bacteria is peptidoglycan, meaning the number one molecule we find in the cell wall of bacteria is peptidoglycan. Any question about any of that? All right. So peptidoglycan is in the bacteria cell walls. Peptidoglycan is a polymer of a repeating disaccharide, meaning peptidoglycan is made up of two sugars, NAG and NAM. Let me blow this up so you can see it better. NAG is N-acetylglucosamine, and NAM is N-acetylmuramic acid. I don't care which name you know, you just need to know one, N-acetylglucosamine or NAG, or N-acetylmuramic acid or NAM, because both of these probably mean the same to you. Mean the same as NAG and N-acetylglucosamine. You have to be a chemist to know what N-acetylglucosamine is. Anyways, here's the sugar, NAG, repeated by NAM, followed by NAG, followed by NAM. You just have that repeating one after the other. And then they line up the whole chain. And they're next to another chain of NAM and NAG, next to another chain of NAG and NAM. These chains are held together by um, short peptides, polypeptides. Here you can see this peptide holding this chain here to that chain there. And there it is right there. That's a chain of 1, 2, 3, 4 peptides. And then this chain is linked to this chain here by this short peptide here. And you'll note that this peptide actually holds on to that peptide here and this peptide here. It's not actually in between this chain of nag and nam and that chain of nag and nam. It's between this peptide here and that peptide there. But the point is the peptides do hold the nag and nam together, and they're held together one chain on top of the other, and then one chain next to the other. So that's what polypeptide, peptidoglycan is. Nag and nam, which are glycans, where the name peptidoglycan comes from, and then they have a peptide linking the glycans together, and that's where we get peptidoglycan from, the name peptidoglycan. Penicillin actually inhibits the lattice formation. Penicillin will get into a growing cell wall, that meaning the cell wall is putting down the nag and nam chains and then attaching the peptides to link them together. Penicillin will get in right here and then prevent that peptide from forming, holding the cell wall together right here. That will create a hole in the cell wall right here. That hole can allow the cell membrane to protrude out of the hole of the cell wall. And then water will move into the cell. That protrusion will get bigger, sort of like blowing up a balloon in a box with a hole in it. The balloon will start coming out of the hole in the box. And as you keep blowing or more water moves into the cell, that cytoplasm will extend and protrude further outside this hole in the cell wall until the plasma membrane bursts or the balloon pops, and then the cytoplasm can then leak out of the cell wall. So that's how penicillin actually damages cells. It doesn't directly kill the cell, it prevents the peptides from forming in peptidoglycan, and then the cell will have a hole in it, and then the cell membrane can protrude out of that hole, and as water moves into the cell, that whole protrusion through the hole will get bigger until the cell will burst. Any question about any of that? All right, we have some time here, so let me talk about the gram-positive cell wall. Has a thick layer of peptidoglycan outside of the cell membrane, shown in yellow here. And remember, it's more than 3 layers. We're only showing you 3 layers. It's actually 30 to hundreds of layers of peptidoglycan thick. The main molecule is peptidoglycan, shown here, the nag and nam, hooked together, the different chains by short peptides, which are these blue things here, linking this chain to that chain and that chain to that chain. I don't see the blue there. Maybe it's those ones there. Yeah, it's those light green ones there. So peptidoglycan is the main molecule in the cell wall. It's not the only molecule in the gram-positive cell wall, though. There are other molecules like teichoic acid and lipoteichoic acid. Teichoic acid is thought to help regulate the movement of cations down the teichoic acid and then through the cell wall and then bring the cation to the cell. However, a cation does not require teichoic acid because a cation will dissolve in water. Water will go right through the cell wall, meaning the gram-positive cell wall. Any molecule dissolved in water will go right through the cell wall. And so the cation doesn't require the teichoic acid. It just helps regulate the cation to move down and then into the cell. We also have lipoteichoic acid, so it has the teichoic acid part, which is helping to hold the peptidoglycan together, meaning it helps anchor the different layers of peptidoglycan together. And then lipoteichoic acid has the same teichoic acid helping to hold the peptidoglycan layers together, but lipoteichoic acid has a lipid on one end of it, and then that lipid can go into the cell membrane. Oh, there we go. So there's the lipid portion of the lipoteichoic acid, and it allows the lipoteichoic acid to attach to the cell membrane and then anchor it to the cell membrane, anchoring the cell wall to the cell membrane in reality. Any question about any of that? The polysaccharides on the lipoteichoic acid and the teichoic acid can provide antigenic variation of the cell wall. Remember, an antigen is a molecule or something, a surface molecule of a cell, which the immune system responds to. And this teichoic acid or lipoteichoic acid can be an antigen, so that if a cell gets infected by this cell, or I should say, a patient or somebody gets infected by this cell, the teichoic acid can be what the immune system will recognize as foreign and then respond to, making an antibody against it. Any question about any of that? All right, the gram-negative cell wall differs in that it has a lipid layer, a peptide, which is the cell membrane, a thinner peptidoglycan layer, and then another lipid layer, which is the outer membrane of the cell wall. The gram-negative bacterial cell wall is very different than the gram-positive. The main molecule is peptidoglycan, but there's much less peptidoglycan in the gram-negative cell than in the gram-positive cell. And they have a whole bunch of other molecules, particularly the lipid bilayer of the outer membrane of the cell wall. The lipid bilayer has two layers to the bilayer. The lower layer right here is very much the same as the bilayer of the cytoplasm or the cell membrane, and that is its phospholipid is the main molecule of the lower layer of the outer membrane of the cell wall. But the upper layer of the bilayer does have phospholipid. In fact, it's probably the majority, but it does have another lipid, lipid A, which is found in the upper layer of the outer membrane of the cell wall, or the upper layer of the bilayer. Lipid A is an endotoxin. So any cell or any patient that gets infected with a gram-negative cell that has a cell wall will be exposed to an endotoxin or lipid A. Any question about any of that? So lipid A is important because it is an endotoxin. Lipid A is attached to an old polysaccharide, shown here, the little hairs shown attached to the lipid A, the big blob in the outer membrane. and that makes the lipid A O polysaccharide a lipopolysaccharide. Altogether, they form a bilayer, which is the outer membrane of the cell wall. Any questions about any of that? Now, there are other molecules in the cell wall, like here's a protein shown here, spanning the outer membrane. In this case, it's also spanning the cell wall. There's also a porin protein, which is a channel, a protein that makes a channel in the outer membrane of the cell wall. The porin proteins are very important because if the cell wants a molecule to enter the cell, it has to enter the outer membrane of the cell wall through the porin protein, and that could be like an amino acid or glucose. The cell's going to want that, and so it has to move through the porin protein. And we call it a channel protein because it allows many different molecules to cross the outer membrane. That differs from the amino acid or the glucose moving through the cell membrane. What we have here is a transport protein that moves one molecule across the cell membrane. So we have a transport protein to move glucose across the cell membrane. We have a different transport protein to move the amino acid lysine across the cell membrane. But both the lysine and the glucose cross the outer membrane through the porin protein. Any question about any of that? We also have phospholipids in the outer membrane of the cell wall or the cell wall. Excuse me, lipoprotein is what I meant. A lipid protein spanning the cell wall, with the lipid portion usually being in the outer membrane, but it could be the lipid portion actually will join the cell membrane, and then the phospholipid portion will be in the cell wall, meaning the peptidoglycan. And there are other proteins that span the cell wall as well. Any question about any of that? Let me see. You don't really need to know the periplasm. That's the region of the cell wall where the peptidoglycan is. You should know lipid A is an endotoxin and it's found in only in gram-negative cells if they have well, I should say gram-negative bacteria cells if they have a cell wall. Lipid A is not found in archaea. It's not found in eukaryotes as well. All right, any question about the gram-negative cell wall? How are we doing on time here? All right. I think it's time for me to end this lesson, and I'll see you at 6:30 in the lab. Are there any questions before I go? All
My
right.
only question, will chapter 3 slides, since we finished them, be posted today so that we can use them for the quiz tomorrow?
Yes, thank you for the reminder. I'll post them right now.
Okay, thank you.
All right. Bye.